Yeah, thank you so much, for the kind introduction, Dr. Langer. I hope you can see the screen and yeah. So thank you very much, Rakesh, for the wonderful invitation. I would also like to thank Dr. Rukma, Dr. Nisha, and most importantly, Professor Manoj there, whom I have known for almost 14 years. I've been visiting School of Biotechnology now and then, for teaching these particular courses. And I would like to thank Vikas as well, for the kind introduction of me to Rakesh, through which this workshop is really catered now. So along with Sundarda and Ashok and myself, we'll take you to these wonderful sessions during the next course of time, the 7 days. We have already given this particular link for you to answer the pre-evaluation. So those who have not answered, you're all requested to, fill up those particular pre-evaluation things. The link is, still open. That will probably allow us, to come up with some rigorous exercises. So out of about 59 participants who have responded for this pre-evaluation, we understand that, there are many students, who are really interested to look into a very basic understanding of bioinformatics. But, although the workshop is for 5 to 7 p.m., we would like you to, ask a lot of questions and come up with any kind of, things that you really wanted to solve. Because this course is more related to systems biology, and bioinformatics necessitated systems biology or systems genomics. So please, feel free to, stop us. So, this is what, we'll probably learn during the next 7 days. So we will come up with very elements of bioinformatics and how, we are moving towards systems biology approaches. We'll largely focus on something called protein-protein interactions. You'll also learn, how these particular data is really fed into public databases, and through what kind of experiments, these are all enabled. That would probably allow us to identify, the niches specific to some of the methods and as well as assays. We'll also learn about the protein-protein interactions, and I believe Ashok would probably largely focus on the cytoscape. Probably, we will use, Osprey and other tools as well to understand some of the protein-protein interactions. So in between, Sundarda will give you a wonderful gist about the databases, the hub of big data, and he will also try to give you, some kind of exercises, and we'll do some kind of exercises now and then as well. So you can use this particular workshop in such a way that any text that is available in red font, please consider them as important. The most important thing is at the end of the talk, you are spending a lot of time for these workshops. Please ask questions. Please don't stop, that curiosity niche. So please keep asking questions, whatever you have in your mind. So this is why, we are here. Okay. Sundarda is in Singapore time, is 2.5 hours ahead of us. Okay. So he would probably, be there all through the Singapore time. So we're all here, to help you, interact and answer some kind of questions. So these are all our email addresses. So if you, please feel free to ask any questions. You're around the clock. I think WhatsApp group is already created by Rakesh. So please ensure that it is done. Okay. So I come from Birla Institute of Scientific Research, which is a not-for-profit research institute. Ours is a Birla Institute of Technology, Ranchi-based not-for-profit institute. Our current chairman is C.K. Birla, who is seen at the end of the figure. So we have two main departments, biotechnology and bioinformatics. The second one is the remote sensing, through which we collaborate and we try to identify flora and fauna, taking the geospatial satellite images. We have the biggest auditorium, and we have got about 14 scientists, about 7 PhD fellows and 2 postdocs, out of which 6 PhD fellows and 1 postdoc is working with me. So ours is headed by Dr. Krishna Mohan, who is at R&D, and Dr. Punendra Ghosh, who is the director of the institute. So whenever we talk about bioinformatics, one of the foremost things that we always should ask is, bioinformatics has really moved up the value chain. Okay, so whenever we talk about bioinformatics, it's nothing but a tool. So, whenever we consider it as a tool, it's just like, ensuring that this tool, could be optimized for your biological understanding. So that is, an absolute entity. And since, we're all biologists, since, most of us are all biologists, we needed, that particular tool to disseminate, the biological understanding or the kind of, things that we really wanted to learn. So if you really talk about a system, any kind of system, might be might hit two components. One is the major components, which we absolutely see, with our naked eye. Like for example, if you take about humans, we always talk about the major components like eyes, legs, hands, the head, and all those kind of things. So these are all the major components. So these major components constitute, tiny entities, which we call it as minor components. And these minor components are most importantly interconnected to each other, just like the major components. Okay. So if you really take an analogy, the most important thing is nothing but the so-called the vehicles. So if you really take a big vehicle like car has got a small tiny parts called vehicles, which in turn vehicles, which these vehicles has got, internal nuts and bolts, that are connected to each other. So these are all the minor components, which make up the major components. So likewise, the human biology is such a way that there is always an interface between the major components and the minor components. Okay. So given this particular understanding, given this particular adage, one of the foremost things that we could ask is why and how these particular components are interconnected to each other. What happens, if one particular connection is given a miss? What happens to that particular entity? Okay, so that's where this systems biology has really come up into threshold. So it's nothing but the biology of a system. So we are trying to understand a dry component called system, in which there are small components, the major or the minor components in the form of the components. Okay, so given this particular organismal level of understanding, so there are always mutations arising and falling. There are always components, small molecules or biomolecules in the form of DNA, RNA, and protein. Okay, they evolute and devolute. There are always these biomolecules that keep interacting with each other. Okay, there's always a protein that might always interact with a protein, a DNA that always interact with the DNA. Okay, if we really take the smaller organisms, by and large, like bacteria, there are various strains that keep adapting and unfitting in the environment. Okay, the best is the case of SARS-CoV-2, the erstwhile SARS-CoV-1 in 2004, that was that came through. It has slightly mutated, and there has been a good number of vectors it has proceeded through, and a kind of a new variant has really come up into picture, which we call it as COVID-19 or SARS-CoV-2. So there are some changes. There are some changes that we often find within the organismal entities. Okay, but these organismal entities could be best discovered using the systems understanding. And that systems understanding is nothing but this biology of a system. Okay, so in that particular process, through generations, there are a lot of genes that are lost and found, and as a result, new species arise, and that is how the interactions keep interacting with each other. Okay, I mean the organisms keep interacting with each other. Okay, so basically, whenever we talk about the systems aspect. There are always contexts in the systems. Okay, so these contexts are nothing but ohms. Okay, whenever we say ohm is nothing but many, okay, or money. So whenever you say genomics, it's a study of many number of genes. Okay, gen plus ohm, it's nothing but study of many number of genes. Okay, likewise, if you talk about proteomes, it's nothing but study of many number of proteins. Okay, likewise transcriptomics, likewise metabonomics, okay, several things. So every organismal entity has got its own system, and its own system is always compounded with these particular entities. Okay, so one of the foremost important things that I can take in case of humans is nothing but, we always talk about epigenetics, which means how probably we as humans, behave, keeping in view of the environment. That is what we call it as epigenetics, or the influence of the environment on how we sustain, or probably how we thrive. Okay. And there are certain defined variables, and probably with these defined variables, we can come up with a lot of phenotypes and diseased phenotypes. Likewise, if you really take plants, seldom, we always talk about what do you call, interactions over there, but these interactions are always, giving rise to some kind of, traits in case of plants. So how good, a kind of a particular plant, can tolerate a stress, a kind of a stress which probably, it is influenced with, whereas that kind of, stress, might give a kind of a completely negative effect if that particular plant grows in different conditions. Okay. So this is what we call it as a quantitative trait loci. So, a QTL or a gene is always necessitated in plants, and that will probably, give us, a good understanding. And that's how, the interactions in plants, keep rising. In case of animals, again, you also talk about traits, how good, the animals, keep feeding themselves. With that particular feeding, can we always talk about any kind of, epigenetics influence? So the best example is, why is that, certain animals or sheep would have a lot of fur if they're really grown in, say, extreme cold conditions, whereas that particular fur is lost when they're really grown in, some kind of, these particular conditions like Indian conditions or summer conditions or something like that. Okay. So I was told, I just got a message that I'm not audible. Is my voice not audible?
So it is audible.
Yeah. Yasir Parvez, I'm sorry, it's audible. I just got a message, but I think it's audible for others. I'm sorry for this. Yeah. Okay. Yeah. So given this particular understanding, so what I was just trying to tell you is at any point of time, you always talk about the major components and the minor components always keep interacting with each other. So in case of bacteria, you always talk about isolates, the pathogenicity, whether or not a bacterial entity is pathogenic or non-pathogenic, okay, and the host interfaces and other stuff. Okay? And the common variable is nothing but that environment. Okay, so if you really see. So this context can always be taken into a pure statistical threshold, which we call it as true positive, true negative, false positives, and false negatives. Okay. So it's nothing but, true positive would be those particular components where you always talk about a real sense. False positive is that particular thing, very impromptu, you'll not really get that particular kind of traits that are associated. Likewise, false negative and true negatives. So given this particular data, given this particular organisms, that herial data, at any given point of time, there is always a multi-omic data, that is being, derived every time. This multi-omic data could be a genomic data, a transcriptomic data, a proteomic data, and all those kind of things. And this various data must be put up in various, gene banks. And these gene banks, would allow us to utilize those particular data sets, and then we can come up with some kind of a prediction models saying that what would be the perchance that given these particular conditions, what would happen if our data is also resembling this particular data. So that's how this bioinformatics has really moved up the value chain. So we talk bioinformatics as a tool, whereas a computational biology as a discipline. But if we really talk in real sense, this bioinformatics enabled computational biology is always necessitated with a system. That system is nothing but an organismal system. Okay. And the genomic component associated with this system is nothing but systems genomics. Okay. So we can, probably, produce a lot of data, from these particular things. So, for our understanding, most of our talks, we always talk about humans, for this workshop. I'm also a human biologist like many of you, but, we can also take some classical examples, whenever we do exercises. So if you really talk about human genomes, we know it's a 3.2 billion base pairs for a haploid data set. For a diploid, it's about 6.4. Okay. And each of these particular cells has got a nucleus within that. So there are about 1 trillion cells in a human body. So each of these trillion cells has got a nucleus, and nucleus has got a nucleoid in the form of a chromatin. Chromatin is made up of chromosomes, which is intercoiled several times, and a bit of that particular chromosomal entity is nothing but a DNA. Okay, and a bit of DNA is nothing but a gene. Okay, and gene transcribes to mRNA, mRNA translates to protein. That is well known, right from a basic, central dogma of molecular biology. But the central dogma of bioinformatics is in such a way that given all these particular biomolecules, there are a lot of sequences, be it a DNA sequence, RNA sequence, or a protein sequence. Okay, so given these particular sequences, can we come up with a kind of a function, be it a DNA sequence, RNA sequence, or a protein sequence? I always give in a classical example, to my students. Take a classical example of some, pearls or moti, So given that you have got a lot of, motia, put up in your desk, you don't know what to do with those particular pearl beads or moti. So what you would probably do is you would probably insert a small thread within those particular pearl beads, and then imagine yourself that you have come up with a sequence, and now that particular pearl beads can be used as a kind of an ornament. Okay, something like a bracelet or something like a necklace or something like that. So imagine each of those particular bases as A's, T's, C's, and G's. Okay. Likewise, if you talk about the protein sequence, imagine each of those entities as 20 amino acids. Now, if you assemble them in an order, that particular entity is nothing but making it as a kind of a structure, and that is how you can come up with a function. Okay, so sequence predicts structure, structure predicts function is nothing but central dogma of bioinformatics. Okay, so that would probably allow you to come up or necessitate a kind of a function given any kind of a sequence, and that particular sequence might be a DNA, RNA, or protein. And in the current cases, it could even be the non-coding sequences as well. Okay, so in a nutshell, the take-home message for this is sequence predicts structure, structure predicts function is the central dogma of bioinformatics. And this is how every cell contains those particular genes. So there are about 28,000 and odd human genes. They correspond to about 10,000 proteins per cell. They might be differentially expressed at any given condition. They might always be associated with different conditions. And they correspond to about 1,000 pathways per cells. Okay. So at the end of the day, given this particular sequence predicts structure, predicts function, you always talk about pathways or ontology. And that is where, you'll have, different kinds of interactions at any given point of time. There is also something called spatio-temporal interactions, which we'll probably go in detail. So at any given point of time, as I told you, in a system, a particular organism thriving in one environmental condition might need not really harbor a similar set of genes during that particular condition. So if the environment changes, there might be different genes that might be expressed in that particular condition. So if there are different genes, then entire structure changes, and if the entire structure changes, then the entire function changes, and if the entire function of that particular cell changes, the entire pathway changes. Okay, so this is what I was just trying to tell you. So in a system, we always talk about major and minor components. Biology is the study of all living organisms. So Kenneth Darwin's hypothesis. We always talk about integrating all the interactions within this particular system, and of course, which survives and competes is always a kind of a best theory that is already exploited. Okay, so there's always an interplay within them, and of course, when we talk about systems biology, it was once considered to be existing, but it's also lost into oblivion. That's because when there are so many interactions that are interacting with each other, there's always a chance that the interactions would always give rise to different functions. Okay, and if there are functions, new functions, and the organisms also would come up in a novel way of evolutionary mechanisms. Okay, So there are new strains. So all these things are nothing but systems biology. So if you really see, one of the most important things of bioinformatics enabled systems biology stems from the good old Charles Darwin's Origin of Species, right, from 1859. Okay, and of course you know how in 1944 every McCarthy considered DNA as a genetic material. Then we could get the first structure of DNA. The insulin structure, the protein insulin structure was has been obtained. And of course, in 2001, the Human Genome Project came into picture. We have been celebrating 20 years of coming of human genome. There has been a lot of personal genomics studies that have been coming up, and we were also discussing about how so many genes do exist across different organisms, different proteins, different genes, and these particular genes and different particular proteins may have probably similar functions across different organisms. But at any given point of time, a novel genome might have novel functions, and some of those particular genes or functions of those particular proteins may not be discovered. So that is where the hypothetical proteins come into picture. Okay. So this is how we come. And today we also talk about the protein expression or gene expression. And in 2013, a wonderful discovery came into picture that many of these particular organisms also have so-called junk DNA, okay, which do not code for the protein. The so-called coding for protein it doesn't entail any coding for the protein, but that might also allow us, to understand what are those particular protein functions. Which means there are some non-coding RNAs which come from the intronic chunks. So about 98.2% of the human genome is, say, junk, was believed to be junk, but it is known that a genetic variation is sitting in the human genome, which might give an essential function as well. Okay, so that's where this non-coding RNAs come into picture. This also plays a very critical role. So the major components include cells, tissues, organelles, organs, whereas the minor components include the genes, proteins, enzymes, the metabolites, and all those kind of things. So they keep interacting. So it's always the dry system in the wet biology. It always involves circuit diagrams for biological networks. There's always a rigorous interconnection. Whenever a given cell is exploited, whenever we wanted to know how this particular cell comes up with a function, we can probably do a single cell RNA-seq these days. I mean, there are two things like bulk-seq and as well as single cell RNA-seq. A bulk RNA-seq is nothing but identifying the gene expression across the entire compendium of all the cells that are present in one organism. Whereas in a single cell, you can try to identify what are those particular changes, structural changes related to single cell. That would probably allow you to come up with a precision scale models, where the current the current research is ongoing. Okay. So we always we don't always talk about the survival of the fittest anymore, but we always talk about the arrival of the fittest. How come the mutant strains keep evolving? Okay, the best example is the SARS-CoV-2. This is one of my favorite slides, which we have hypothesized some years ago. So as you could see here, genomics is nothing but study of many number of genes, right? So whenever we talk about genes, it's nothing but the whole compendium of the human genome. Okay, so it's about 6.4 billion base pairs of a deployed data set. Okay, so it's about 3.2 GB of data set, human genome. So that is a genome, there is a human genome. Okay. Now, this particular 3.2 GB of human genome size translates to several proteins. So if you really take this genomic component, including the coding and the non-coding, there are many expressible proteins. So that's one of the reasons why the size of the proteomics here is bit large. Okay, so genomics forms a base, it's 1x. Maybe proteomics could be 3x, maybe 3 times larger than the genome. Okay, and you've got a lot of metabolites that are interacting with each other, the physical and as well as the chemical metabolites that are associated with that particular organism. Okay, that is what we call it as metabolomics and metabonomics. Metabolome is nothing but study of many number of metabolites. Metabonomics is nothing but study of many number of these metabolites, heralding a kind of a known function to that particular protein. Okay, this is small difference. And these metabolites, they keep, interacting, but not all metabolites, give rise to a proper function. So here the interactomics, here you get a lot of metabolites again, something like the proteins. Okay, so they keep interacting with each other, but not all interactions give rise to functions. So basically, the size of the genome is equivalent to the size of the function. Okay, so which means the functomics is nothing but equivalent to genomics. So it's nothing but, in a principle, in a theory, if there are some X number of genes present in that particular organism, in principle, those number of genes would necessitate similar function or similar sized function for that particular organism. Okay, so this is how this particular hypothesis is set up. There are different omics existing these days. There are about 339 omics existing these days. We have 0.2 omics. One is synthetome, which is a synthetic interactions. Okay, synthetic protein interactions. The other one is hypothome. Along with Sundarda, we have hypothesized, which is nothing but interactome of hypothetical proteins. Okay, so there are a good number of databases, and these particular databases would really allow you to identify these omics existing these days. Okay, but whenever we talk about systems biology, again, please bear it in mind. Whenever we talk about systems biology, it's always the bioinformatics necessitated systems biology. Okay, and likewise, there are two types of systems approaches. One is a kind of a dynamic approach, where dynamicists always talk about pure systems theory. Someone like a pure bioinformaticist or a pure computational theoreticist. Okay, who would only simply think that very good at theory and he wanted to pursue that particular theoretical knowledge in deriving the function of those particular proteins or genes. Okay, and these are basically based on models or networks. Okay, so these are either quantitative or qualitative. Whenever we talk about quantitative models, there could be many number of genes, and those particular genes can be modeled, and not all genes can give rise to a proper function. So at the end of the day, the quantitative models will really give rise to some qualitative models. Okay, we're only talking about few of these genes or proteins that are necessitated functions. Okay, there are reductionists like many of you. Okay, who would probably want to come up and say that yes, we wanted to come up with bioinformatics predictions. Okay, like probably you and me, and say that yes, let's predict bioinformatics based approaches and come up with a molecular biology experiment. Okay, and that is simply based on something called the high throughput models. Okay, so this is what Hugh Angus has wonderfully proposed, and this always is in the realm of understanding bioinformatics or biology or systems biology. Okay, so we always come up with bioinformatics predictions, take these predictions to the wet lab and come up with an experiment. So that is the beauty of taking the systems biology experiments. Okay, so I just wanted to have a quick break. Do you have any questions, please? Yeah, anyone, please? If there are any questions, I'll probably go to the next part. I'll stop here, or maybe I'll so assuming that you're all yeah, slide number 19, I'll definitely go there. So assuming that you're all good listeners, I'm probably going to my part. Okay, yeah. So sorry, 1 minute. Yeah. Okay, so don't worry, I'll share these particular slides with you. Okay, so please don't write any of these slides. Okay, so the loose definition of systems biology is application to projects, okay, exploring individual biological networks, and the broad text derived definition is outgrowth of these theoretical models, taking up systems theory, okay, engineering, mathematics, statistics, and artificial intelligence. So it's a conglomerate of various disciplines. That's where the systems biology come into picture. So as I told you, it always entails the omic data. So it could be the gene expression data, the metabolomic data, the metabonomic data, the phenomic data, which involves the phenotype data. Okay. And of course, the interaction data, which involves both pathways, quantitative and qualitative models. Okay. But over the last few years, one of the foremost things that we need to ask here is, systems biology or bioinformatics enabled systems biology has grown, by and large. There are many bioinformaticists who are also doing experimental biology as well, or many experimental biologists like you who are doing bioinformatics predictions in their particular work and experimenting in the lab. So now a big, cohort has come up. Now this cohort is really, making a lot of, discussions. So one of these particular discussions is, how do we know, okay, that if at all, if there are interactions that are prevailing in a system, how do we know that some of these interactions are bona fide? When I say bona fide, they are genuine. Okay. How do we know, okay, that these interactions are genuine? Okay. So in basically what is in theory, so what is that dynamic approach that we were discussing earlier, where we come up with a model, it need not really fit practically. Okay, that's because many reductionists think that this is a pure theoretical prediction, this is a pure bioinformatics prediction. Unless otherwise you experiment in the lab, you can't say that it's a final goal set. Okay, so that's where a hypothesis-driven approach need not always be an experimental-driven. Okay, so if there are interactions, can we take measure of unknown interactions in a system? Okay, it's just like a transition property. Say in a room, if say for example, if there are three people, if A interacts with say B, okay, and B interacts with C separately. So B is a common man. Necessarily, it will be very easy for A to interact with C through B. Okay, it's just like a normal Facebook connection or a LinkedIn connection, so that you can get connected. So we can also identify the function of those unknown interactions as well. Okay. So through this understanding, I think, systems biology has really, grown up of ages. So there are various definitions, these cohort have really come up with. So one is a molecular biology is nothing but study of biology at molecular level. Computational biology is how these particular molecules work in silico. Molecular systems biology is nothing but making these molecules work in a system. Whereas a computational systems biology is making these molecules work in a system using in silico approaches. Okay, so there is a cohesive need to define these particular disciplines in a nutshell. Okay. So at the end of the day, as I told you, if in a room, if there are several people interacting with each other, there's always an organization. Say in a room, like in a classroom, how we sit, the proteins that are interacting with each other can also be having a good organization. Okay, please bear it in mind. In systems biology or in bioinformatics, we largely talk about proteins because they are the expressible entities. Okay, whereas the genes form a smaller cohort. Okay, but largely in systems biology we talk about proteins because they are the ones which will allow you to define the phenotype of interest, which will allow you to understand whether or not it is really causal to that particular disease. It will allow you to understand whether or not it is causal to that particular condition, which we were talking in the earlier slides. Okay, so there's always organization, there's always cooperability, there's always mapping, and there are again two types of systems biology approach. One is the top-down systems biology, and the other one is the bottom-up systems biology. So in a top-down systems biology, DNA transcripts mRNA translates to protein. And now you talk about proteins and simply ask what are these particular doing in your cell? Are they interacting with other proteins sitting in the cell? Okay, and now if they are interacting, say protein A with one type of function is interacting with say protein B like this. Okay, then I can say that protein B function, if it is unknown, it can be taken up through protein A. So whatever protein A does, maybe protein B can take that particular function from protein B. Okay, so that is how we identify the function of these particular proteins. Whereas in a bottom-up systems biology approach, what are the proteins already, what are the functions that are already. Now you go up. Now you ask, what are these metabolites that these particular proteins are associated with? Now these metabolites, do they have any regulatory association with the components sitting in the system? Okay, do these keep interacting with each other? What happens if there is a flux imbalance in the cell? What happens if there are fewer number of proteins in one cell and more number of metabolites in one cell? What happens to that particular cell, or what happens to that particular organism by and large? Okay, so this is what we call it as bottom up systems biology, and that's where this human protein interaction networks came into picture. So then now you might want to ask, Prash, Apitna, good scenario, are there any kind of protein interaction databases, or do we have the entire compendium of organismal protein interaction, for various organisms? So in case of humans, yes, we already had first compendium in 2014, which was advanced, which came up, with 2020 and 2021 as well. There are a good number of protein-protein interaction databases and as well as visualizers for different organisms of interest, which we will do as a part of our exercises. Okay. So my 16 to 18 years of research career has really gone through this particular phase. Okay. So what happens? Say this is a protein of interest. Okay. So this is a repair gene, which is at the center over here. Now what happens if this gene is interacting with several genes? Okay, I can say that these are all the genes that are associated with DNA repair as well. But I find a protein from my predictions, from my bioinformatics prediction, that this is not really known. This function of this particular protein is not known. Now, can I say that this hypothetical guy, this hypothetical protein function, can be taken up by this particular protein? Okay. So this is how you can come up with a hypothesis saying that yes, two proteins are physically interacting. Okay, if by virtue of these particular interactions, you may say that they are physically interacting, and I can come up with a kind of a function. So if you really look back, this is nothing but a very basic understanding from our undergraduate biochemistry, which we know from the so-called the lock and key model. I think all of you know what is this lock and key model. Okay, so if you really see, enzymes as proteins, okay, they recognize specific targets and bind them. That is because that specificity, that specific target specificity is more related to conserved areas or those particular niche areas. So, for example, if this were a protein, it would only find something like a lock and key model like this. Okay, so it will probably fit like this. It may not really. If this is like this, if there is another protein like this, it may not really fit. Okay, so it's always like this, or maybe like this, or like this. Okay, so that's where the specificity results in conservation, and that's where you can define the structural properties or the physicochemical properties for that particular function. Okay, so that's where you can talk about. So in systems biology, we always talk about known interactions. You can also state the function. Okay. You can also come up with computational models or systems biology models. You can come up with homology-based models or interaction-based models. That's where you can define the interactability of that particular protein, and you can come up with those particular functions. And based on the type of interactions, there are different kinds of entities. Say if the interactions are similar, then those interactions are like homogeneous interactions. Like for example, in organism A, if two proteins are interacting like this, okay, in organism B as well, if even though you don't find those particular proteins, you may say that those particular proteins are interacting like this only, because they are in fact the same proteins. They are nothing but the orthologous proteins. Okay, they're nothing but the similar proteins, right? So similar proteins across different organisms are nothing but orthologs. Okay, likewise, heterogeneous interactions. Maybe not necessarily similar proteins might always interact in a similar way. Usually they interact in a similar way, but there might be different phosphorylases or kinases. Maybe they might interact differently if they go to a different cell. Okay, so those particular interactions are called as heterogeneous interactions. Okay, and likewise, there are organism-specific interactions. Maybe in one kind of organisms, maybe in bacteria, if we have one kind of protein, they might be, interacting in one way in that particular organism. Whereas if you really look into a completely different eutherian in a different, say, eukaryotic organism, maybe they might have different interacting patterns. Okay. Likewise, we also have identity specific. Identity specific is something like this. You see, यहाँ पर, say in one organism, this is your identity, this is your core protein, and the other proteins that are surrounding to it are called as the hypercore proteins. If you really take this knowledge and try to ask whether or not the interactions are quite similar across different organisms, then they might be different across different organisms. Okay, so that is the difference between a core protein and a hypercore protein. Okay, so we always talk about these interactions at the regulatory scale. Regulatory scale is nothing but in your window, say this is your DNA of interest or a gene of interest. At the 5' untranslational region of this particular gene, there might be a lot of non-coding RNAs that might be sitting. There might be a lot of enhancers. There might be regulatory elements. There might be transcription factors as well, because this transcription factor defines how this particular gene transcribes to mRNA. Now, what happens is, unless and otherwise, we know what are these particular transcription factors, we will not be able to know the function of these particular genes. Okay, so this is how we can identify how these particular genes behave, so that we can come up with novel strains. This is what we call it as a neo-functionalization. And then we come up with a lot of new genes and proteins and come up with experimental approaches. Okay, and the best example is nothing but taking the phenome interactive network. Phenome is nothing but a study of many number of phenotypes. Okay, it's nothing but phenomes. Okay, so now what happens is, many a times, say for example, you talk about diabetes. About 8% of Indians are diabetic, which is quite alarming. Now, can we say, based on some data from this 8% of Indian population, can we say what is the predictable risk of the other 92% of Indian population, taking the 8% phenotype data that we have, who are already at diabetes risk? Okay, so we know that these 8% people are already diabetes. We have that particular data. We know what are their lifestyle habits, eating habits, okay, their body mass index and other entities. Now, can we say that yes, we have the other 92% or at least other data through which we will try to match that this particular data matches with the already existing diabetic risk score data. Can we come up with that particular predictive analytics? Okay, so that's where the machine learning, the predictive analytics come into picture, where Sundrararajan will give you a wonderful gist about that. Okay, this is all bioinformatics understanding. So there's always functional relationships between these diseases and disease promoting genes, or even the promoters, because you already have a gene, you have the promoters, you have the transcription factors, enhancers, regulatory elements, the open chromatin elements, the transcription factor binding sites. So many things would have an influence on how this particular gene transcribes. Not only that, it also has an influence on how this particular gene probably translates to a protein. Okay, so all these particular things would really come into predictive biology. Okay, so the various applications associated with this, these applications could be better ascertained using the protein-protein interaction data. Okay, so you may say that the protein-protein interactions have really given us a good understanding, because there's always a huge diversity of protein interactions. So for example, if this is a protein, as I told you, the lock and key model, there might be some other groups over here. Maybe the other protein like this can interact as well. So this is already a protein over here. This particular protein interaction has a lot of diversity, because if you really take this particular interaction data in other organism, you might find another protein that might be interacting as well. So now, even if this particular protein interaction fails, you have the other group sitting over here. Okay, so this diversity or this knowledge is really taken up for better understanding the function of the proteins. Okay, so this is where the protein-protein interactions are absolutely applied. So we use different databases, different, experimental data as well. In fact, all this particular interaction data is taken up from the biologically fed data only. So it's only you experimental biologists, okay, who keep this particular data predicted and publish it, and that published data is automatically fed into various databases. So we may say that close homologs interact in the same way. So when I say protein A in one organism and protein A dash, which is an ortholog sitting in other organism, okay, might have similar interacting partners, like A is interacting with B. Likewise, there might also be something like B dash as well. Okay, so similar proteins across different organisms might interact similarly. Okay, that would probably give us a lot of divergence. That would probably give us a lot of understanding towards the function of these particular proteins. Okay, so do you have any questions before I move further? Yeah. Am I going fast, or do you want me to stop anywhere? Yeah. Any questions, please? Yeah. So if there are no questions, I'll probably continue. Yeah. Yeah. So, so this is one of these particular protein interaction maps, that which we have drawn, when there were no visualizers, way back in 2005, I believe. Okay. This was one of those first interaction partners, with with those things. Yeah. Yeah. So Sangeeta, yes, I request all of you to ask questions in not in private mode, so that others also can ask those, see those questions. Please make in public mode. Yeah, I'll take you, through some of these particular protein interaction databases as well. Okay, and how to create a database. All these things, Sundarda will definitely explain. Okay, so this is one of those classical examples of this protein interaction map. You see, all I would say the bold ones are all our interesting proteins, irrespective of the size or shape or whatever. Okay, so which means, as you could say here, this is a bold one, circle one. These are all our proteins. All the yellow ones are all the mitochondrial proteins. Okay, all the red ones are all the diseased proteins. So when I say, for example, this one is our protein, which is a mitochondrial one and also known to be associated with a function. This is what we know. It's a simplest DNA mismatch repair protein interaction network. But this is only the first degree interaction networks, okay? Which means there might be interactions associated with the second degree as well. So just imagine your Facebook or LinkedIn, okay? You will get a lot of friend invitations, okay? Through friend of friend of friend. So your first degree interacting partners are your best interacting partners, because you already know them physically. Okay, you have met them, you have known them. They might be your close family friends as well. So the friend of friend, probably you may not know, but you can rely upon, just because he is connected through your friend. Okay, so that particular interaction could be the second degree interaction networks. Okay? So this is how your second degree interaction networks, which is a very condensed network that comes in. I'm sorry, I'm hearing some voice. Do you have any questions here? Yeah, I'll come up with these things, so please don't worry. So why big names for these particular what do you call proteins? I don't think there are no big names. This is the reason. This is during those days in 2006. As a part of my PhD. There were we don't know, because there were no visualizers at that point of time. I had to write it. Okay, what is that particular function? With a pencil. That is what I've written it. Carefully. Okay, so please. Protein names are all usually short. They're nothing but the protein coding chains. Okay, so these are the second degree interaction networks. So now I could come up with a conclusion saying that this is the guy from my erstwhile query, that these are all the proteins that could be associated with my function. Okay, so this is how I can come up with a question. So the first part of my talk is We should always ask, how these systems biology approaches have really taken into shape. Are protein interactions always biological? Why protein-protein interactions? Can we identify the function of these particular proteins, through interacting partners? In other words, can we say the function of the known, unknown regions or the proteins which does not have any function like the hypothetical proteins? And what are the kinds of databases and tools that we'll be able to know during the exercises? So in systems biology, you always talk about an integration. So integration of various disciplines. It's always multifaceted. Please bear it in mind, it's not systematic biology. Systematic biology is taxonomic biology. So systems biology is theory, systems theory and biology explained. It's always an intersection of number of interactable entities, interactable biomolecules. And of course, you always take a measure of bioinformatics predictions, in silico predictions, and then experiment in the lab. Okay. So having said this, so this systems understanding can really be taken up in a more nutshell. You might want to say that maybe if you want to directly experiment in the lab, maybe you want to identify a region of interest, clonate or PCR, identify the bands or correlate, or maybe come up with a pull down assay. That might allow you to do an experiment, say, in 24 hours, okay? If you're really a workaholic. But if you're a bioinformaticist, the bioinformatic is, or lessen the scale of your experimentation, do it something like this, take the sequence, okay, and see what are those similar proteins, identify the domains, correlate, and then you can come up with a simplest conclusion. All these things will probably take up within 30 minutes. Okay. This may not really be comparable with your experimental work, but at least from larger number of sequences, you can jot down to at least smaller number of sequences at least. Okay, so that's where these particular interactions would really allow you to move forward. So last 10 minutes of my talk, I'll probably try to give you approaches how these particular data is really fed into these databases. Okay, which if you are an experimental biologist, you will probably love this. Okay, so many of these interaction databases, many of the systems data is either fed using the so-called pull down assays. Okay. Pull down assays are nothing but the Western blots, the co-immunoprecipitation, the yeast amino acid, yeast to hybrid screens, okay, or the tandem affinity purification mass spectrometry, the LCMS data, the GCMS data, okay, and so many other things. So all these experimental approaches are nothing but the physical interactions, because you know that those proteins are physically interacting with each other, okay. Whereas the computational approaches, these are all homology driven. They're all the homology based approaches. So they're simply based on the virtual interactions. It is only simply based on the simplest matches that are available in the publicly available databases. Okay, and then you understand the function of genes, and then you build a testable model for these particular cell functions. So there are different experiments, as I told you, like the chromatin immunoprecipitation, the peptide arrays, the microarrays. Nowadays, the next generation sequencing based approaches, like the Illumina short read sequencing, the PacBio, the long read sequencing, the affinity chromatography using mass spectrometry, the yeast to hybrid screen. So these are all the high throughput methods. Okay, and since this workshop is organized by the Department of Genetics, you must be knowing what are the double deletion mutants or the abundance assays. You might really come up with an argument saying that could there be genetic association with your proteins of interest or with genes of interest? Okay, that's where the genetic interactions come into picture. There might be text mining interactions. You simply check from your literature saying that two genes are physically interacting. There might be some literature that might be known in, say, PubMed or something like that. That's where the information hyperlinked over proteins, the HIOP, which is a database, which can give you this understanding. And there are also association networks like String, Gene Mania. Okay, all these protein-protein interaction databases, are really fed. Okay. So you might ask me with one question. Okay, Prash, you always talk about protein-protein interactions. Ain't there any protein DNA interactions or protein RNA interactions? or DNA-DNA interactions. Why is it we're always talking about proteins? Of course, one answer is very obvious. We are always talking about the protein as a translatable entity. That's because that particular protein would have a rigorous knowledge or influence on how that particular disease takes place. Okay, that is very clear. And now, in order to identify the function, we need another biomolecule. So the best molecule to understand this intractability is nothing but another protein, because it speaks the same language. Because if two proteins are physically interacting, then you are talking about two proteins that are associated with a disease. But nevertheless, you might also have a protein like this, where many a time this translatable entity might also interact with the DNA as well, or with the RNA as well, or with the non-coding RNA as well. Okay. So this interaction always gives rise to a regulatory nature. Okay, this is something different. This gives a regulation. You will know what happens to that particular interaction, what happens to that particular cell if a protein interacts with other components as well. Okay, so protein DNA, protein RNA interactions are also there. And for this, yeast to hybrid screens or Western blots are well known. I think many of you wet lab biologists know this. I'll just try to give you a gist of it. So it comes on with a hypothesis that there are transcription factors. These transcription factors influence that particular the way that protein coding gene takes place. You have two major entities. One is a DNA binding domain and an activated domain, and then these two particular entities are really fused with each other like this. And then the functional transcription factor is really created if we know from the expression that an activating domain is known to be interacting with the DNA binding domain. So which means you have a protein of interest, which you call it as a bait protein, for which you raise an antibody. You ask your prey proteins to come and interact with this particular bait, so they keep interacting. So if at all, if there is an interaction, then that interactability or interaction mode is done based on the strong, interaction mode. So if it keeps on interacting like this, it's a strong interacting partner. If just interacts like this and falls, it's a weak interacting partner. It's just like kissing interacting proteins. It's just like something like the kinases or the phosphorylase. Many of these kinases, they just act. They just try to interact and they just chip away. Okay. These involve a lot of novel interactions. They are very much, in vitro based approaches. So this experiment is also called as in vitro transcription translation based approach. Okay. So there are also co-immunoprecipitation based experiments always done like this. So this is how it is done. It is immobilized when protein is there. As I told you, a pull down is nothing but you're pulling down one particular protein as a bait and asking the translatable proteins or the other proteins of interest to check whether or not they are physically interacting with each other. And then you run the gel. And if they are in the similar or parallelly running to each other, then that bait and prey are physically interacting with each other. Okay. So all these things are nothing but the pull down assays. And as I told you, interactions are of two types, the stable and as well as the transient interactions. And of course, mass spectrometry is also well known. It usually allows us to identify small peptide molecules or small metabolites. Okay, we can detect them based on the masses to charge ratio. Okay, there are two well-known mass spectrometry taking into the matter of. One is the matrix-assisted laser desorption ionization time of flight. The other one is cell of the surface enhanced laser desorption ionization time of flight. It will know how many number of proteins are translatable to ions or small peptides, based on which the differential expression can be. Okay. So many of these particular proteins, usually the highly expressed proteins evolve slowly. This is one of those particular favorite papers, which we have discussed earlier. So many of these particular proteins, highly expressed proteins evolve slowly. So if many of these highly expressed proteins evolve slowly, if they evolve slowly, then we may say that the interactable nature is also slow. Okay, so that is how the functions are always defined, and always we may say that interacting proteins always co-express. So which means let's imagine you have a big university like University of Jammu. There are various departments. So maybe Department of Genetics, people might be interacting with each other. They might have similar jargon, understanding when compared to Department of Zoology or Department of or School of Biotechnology. Okay, so it's always that interacting partners always co-express. The other way around is co-express proteins always co-interact with each other. Okay, so that is how it is. So if there are two proteins that are co-expressing in one organelle, say in mitochondria, they always physically interact. Okay, the other way around is if two physically interacting proteins always co-express, it's just like twins, they always come together. Okay, likewise protein microarray has come into picture, and that is how we can identify the functions of these particular proteins. Okay. Probably I'll just stop here since my time is up. But most of these things we'll do some exercises how to come up with protein interactions and all. Most of these are online tools. But I'll stop here for quick questions if you may have. Otherwise, I'll let Sundarda to come up with his wonderful talk. Yes, there are questions, like, could you also educate us about protein RNA interactions? Yes, it also comes under systems approach. There are a good number of tools. We have recently come up with a hypothesis on RNA protein interactions as well, Sheetal. So it also comes into that understanding level. So you can also do those protein-protein interactions, or protein RNA interactions, just like protein-protein interactions. All the protein RNA interactions is just budding, it has just come into picture right now. It is just coming up. Yeah. So any more questions? Yeah. So if there are no questions, probably I'll stop here. Assuming that you all have understood, you are most welcome to post questions in the WhatsApp group. Okay, I'll be happy to answer any questions that you have, and we'll also do some kind of exercises. Many of you are attending. Please do the tree evaluation thing as well. Okay, yeah. So thank you so much. Over to Sundarda. Sundarda is my mentor. I'm sure you'll really love his talk. Please feel free to ask questions from him as well. Yeah. Thank you very much.
Thank you, Prashanth, for the informative talk. If Dr. Vijay is there, we can start the second lecture.
Yeah, just a minute. Let me see whether I can share the screen. Anything visible now?
No, not yet.
Yeah, I think it got disconnected again. So that's why using my phone and also the computer it got disconnected again. Few minutes.
No problem, sir. You can take your time.
Audience had a wonderful thought. Yeah. Is it coming?
Yeah.
Okay. Hi, good evening, all young minds who are ready to take up the bioinformatics as a main area of your research career and job. One thing I want to tell you, even though I also started as a bioinformatics for biology only, okay, and it went on long journey, not only in biological area, but I went through a lot of areas, domains like bank also. We model a lot of things in a bank, in a leading bank in Singapore, and then I worked in anywhere. We made a lot of disease modeling. We made students enhancement, and even you'll be surprised that I used the bioinformatic tools for predicting the vector, dengue vector spread in Singapore also, and it really worked very well. And we could contain it in 2015, where the dengue cases were increasing beyond control. And so if you choose bioinformatics, one thing is sure, whether you are getting a job in biology or not, you will get a job anywhere in the world, in any industry, because all the tools we use in bioinformatics are the basics for all domains, because that's how the bioinformatics have gone in multi-domain. And I'm also doing a lot of work in different domains, and not only in biology, but in every domain named. You can say it is possible to apply the bioinformatic tools and come up with a lot of things. Okay. So far, first of all, I want to thank Professor Rakesh for the nice invitation and the opportunity to talk to so many young minds and master students and the budding faculties in Jammu University and other places. I saw a lot of few participants from outside, also from other countries like Malaysia, South Korea, and other places like I saw some people from even Bangladesh, Nigeria, Egypt, Uzbekistan, and so many places people have joined. And really, it is a very interesting thing that to see that people around the world are more interested in see what is happening and how we are going to use the bioinformatic tools in any domain. Okay. Okay. So for Professor Manoj told, we have mentioned big data, and Prashanth told a lot of data banks, several, almost I think now it is about 1,000 databases which cater for various knowledge and things like that. So then, like Professor Manoj told, we call it as a big data. Yes. So now we are calling it as a big data. And I think it's, are you able to hear me?
Yes, sir.
Yes, sir. Yes.
Yeah.
Yeah.
Yes, sir.
Yes, sir. Can you hear me?
Yes, sir. Yes, sir. Please.
The slide is closed. Yeah, okay. Only the slide. Prashanth, are you there?
Yes, sir. It's clear. Yeah.
Yeah, I sent you the files. Maybe I think it was working a while. Again, it stopped.
Yeah.
So you want to put the PowerPoints? Send it to your email.
Yeah, sure. I'll do that. 1 minute. Yeah. Maybe you can stop sharing the screen. I'll share the screen on your behalf. Yeah.
Yeah. Yeah, I will stop sharing. It has already ended my sharing. It's very strange. In Singapore, we get such connections problem, because now most of the
You may
people
go ahead,
are working
yeah.
from home. So that's the reason I think that. Yeah. Thank you. Yeah. So as Prashanth mentioned, you can see a lot of names here. Whatever name you saw in 50 slides in Prashanth is already put here in one slide. The bioinformatics can go for databases, sequences, and whatnot. So then what is the big data? So big data, sometime back when I used to say, I was accumulating data over my 200 GB. Slowly one day I found I don't have more space. Then I went, nowadays we get 1 terabyte of a disk, or even thumb drive is possible. So where we are going ahead? I mean, is it called big data, or is there something we are doing wrong, or what are we doing? So the thing is, we are start accumulating a lot of things. Even sometimes we accumulate a lot of books. We don't know whether we'll read it or not. So same way, we are getting huge surge of data from various sources, and genomic data or protein data or whatever. So when you combine everything, then we can say it becomes a big data. Means data beyond our human perception to read and analyze or understand and do. We don't know what to do with that finally. So there are labs in the world. They accumulated a lot of data theoretically or experimentally, and finally they don't know what to do. They just write a paper and then, okay, that's the thing they can do at the best. So are we able to do something with all this data, not only in biology or in various domains? Is a question now we are getting. Next slide, please. Prashanth, okay, thank you. Yeah. So just now we were hearing about the human genome projects. As of today, this one is taken from today. See, the cost of human genome, how to find the genome, the cost has reduced quite well. So because of this, every lab has a capability in producing many genomes. So we can see how the cost has reduced from 2007 to 2020, and it's still reducing because of various technologies has come up, and so this is again a big surge of data. I mean, we are getting huge data about various species and plants and whatnot. Anything you name, we are able to model it. We are getting a genome, and even the recent one is a COVID one. Is only 29,000 base pair, but the way the world is shaken out by the 29,000 base pair is beyond our capability to understand how these genes and things are functioning in such it can spread. The entire world is now hidden in home now. So all these things are still under miracle, or we are beyond our knowledge, or we are unable to understand things. So again, we get more data over data. Okay, next slide. So when we talk about a lot of genome, then we ask immediate question, which concerns everyone due to the COVID or any fast spreading diseases. What causes genetic diseases? So when there is a change in DNA, part of the DNA, then the mutation occurs. This all you all know because you are studying on that. So this causes ultimately to get some disease or a problem in somebody's life. So the mutation, even in COVID, was multiple mutations, and we say South African strain or UK strain. Again, India also got a new strain, and so there are so many strains coming out. In Japan also they got a new strain. So what happened? The same 29,000 suddenly got some changes. It changed itself while evolving to a new person or getting for a new infection. So this causes a problem worldwide. Are we able to do something? I mean, are we able to predict whether a genome or gene is going to have a mutation or not? It's a very big question, which we are yet to know whether it is going to happen or not. So this is the reason that diseases are spreading, and medical biologists and pharmacology companies are dying out in finding out the vaccine or medicine for all these things. Okay, next slide. So now comes the question, bioinformatics and big data. So bioinformatics with data analytics tools. What is data analytics tools? Around this 7-day workshop, you'll be able to see some glimpse of what is the tool we are going to use and what things are happening with the huge data like this. So if we use the data analytics tools, it will ease our understanding on the huge volume of data that were collected or that were publicly available over the internet or all the places. So what is the use of this? So is big data available, and what are the bioinformatics tools we are able to use it to mine or bring out the hidden information from the big data? So we can do the identification and treatment of a disease at a faster scale. Because if we know the mapping of disease to genome in a wider spectrum with more enhanced databases and clinical trials, then we are able to make a quicker treatment of diseases in a faster scale. And cost is less because we are going to do models only silico, not in the real. So silico means only the buying a laptop, or even a laptop nowadays is capable of running a big scale experimental theoretical experiments. So cost has reduced significantly, and better health is provided at an early stage, because you can see how COVID vaccination has come out very fast because of a lot of things behind silico. And care of patient is moderated to regular data updates. Means because of data in silico, we are able to update worldwide all the clinical trials and things like that. And population disease control measures are ignited. Means we are able to control the disease spread quickly, and then we are able to contain the disease within a country or within a region. COVID is a very classic example how we are getting into trouble and how things are controlled in most of the countries. Okay, next slide. Okay, data and analytics. So now comes to now we know we got the big data, we got from various sources. So what data we got it, and then what we are going to do with the analytics. So data we are collecting in bioinformatics or biology field, DNA sequence or genomes, amino acid sequence of protein or 3-dimensional structures, and various complexes and whatnot. These I am naming as a few. So we already saw that because of genome projects, we got a huge number of genomic sequences available. So what data analytics we are trying to do? Are we able to use this data partially or fully? And then can we do some prediction, or are we able to mine the data and see whether a particular gene has a protein function? Or can I group set of genes and see experimentally I know that some set of genes able to do some protein function. So are we able to use the data which we have and predict other genes which we didn't know earlier by experimentally, whether it can give possibly direction to say it can do a particular prediction or protein function. So that's the idea. So once we collect all the data and we now aim to see through the data analytics tool, are we able to build model in predicting protein function from gene with the genomic sequence or other available data? Any question? So one is the big data and the other side is the data analytics. These two should be very clear that one side I'm accumulating. Yeah. Any question? Okay, next slide. Okay, what are the impacts of big data in bioinformatics? Let us go by topic. Like first I take only the biology portion. So if I take biology portion, the traditional way of what we used to do in experimentally, but now we are going to do through sophisticated techniques, means the modern technology. What is the difference? Earlier we used to get funds and get some lab oriented in finding a gene or a protein function and things. But now we are doing most of the things in silico. In this, I want to give you a classic example. You know the 9/11 Twin Tower blast in USA. There were 3,000 to 4,000 people died, and they were searching to see who are the people. They could not match. They cannot identify who are all the people together from the whatever remains available. People will not believe. Suddenly, somebody suggested, "Why not you go for a bioinformatics company?" There is a very small company in US. So they approached them. Can you find who are the really people died in that big 9/11? Then they came into picture. They went over all missing. They went to their home, collected from their comb or this or that DNA, and then all the DNA found from the site. They constructed all the DNA and matched and really found who are all the people who really died in the pig. So that time people realized what the power of bioinformatics. And without that, the US, they could not have found who are the really people who died in the major problem. So biology has a very strong use for the bioinformatics in cost saving or time saving. We can do a better analytics, and the big data, we can predict genomic or protein sequencing, and even sequencing. Nowadays, many people use the silico techniques rather than they don't go for a full-fledged sequencing. First, they try to model it silico or through the prediction models, and then they see whether this particular sequence is going to be a new one or a already partially known one. Accordingly, they go for the lab experiment. So it reduces the huge cost, time, and they're able to build a sequence much faster scale. And also, as Prashanth was talking, like prediction of protein structure. So this gives a direction. This is even is a very good thing that if we are able to get the structure, then we are able to solve most of the other problem, getting a targeted drug or anything. And if we do experimentally, is going to be an immense problem, time scale, and huge expense. But because of silico thing, it can be solved in an easier, better, solvable way, and things are moving in that way. Okay, next slide. So what is the impact in gene sequences? So gene sequences, we are able to build genetic code. Earlier, we used to map and get the sequences. And how it is mapped, we are mapping to the human genetics. So how it is going to help with the big data? With the big data, genetic disorders, blueprints can be easily get into. We are able to map the genome. We are yet to get the personal genome. Maybe next decade, we are going to, everyone will have their own genome. They can carry that genome in thumb drive and say, this is my genome. Can you check whether I'm going to get this disease or not? This is not going to be a big imagination, but it's going to happen maybe in next decade. So once we have that, then we know if once any disorder or any disease is mapped already, then we know where I stand, whether I'm going to get it or not, when I'm going to get it. So all these are likely to happen because of the volume of data is increasing. And later, the second part, I'm going to caution you, don't just accumulate data. That's also I'm going to talk in the second portion. So while doing this, we are reducing the cost in sequencing and less error due to analytics algorithm. Because of the strong analytic algorithm, we are able to get better sequences than through other methodology. You know, a lot of time I use, even I used in my PhD microarray data, but which is very biased. One time I get certain data, one gene is highly expressed, one gene is low expressed. Next time I get something different. So it's not uniform. So because I use the real traditional methodology, I always end up in confusion because my method also cannot work because it's bit of biased data happening because of some error, probe error, or whatever error it's going to happen. But if I do it silico, 90% problem is solved because it's a confirmed established data with which I'm able to build and predict the sequences. So which is going to be more error free than the traditional methods. Okay, next slide. Healthcare. This is what everybody is worried about. Are we going to get a good healthcare? Where this big data going to help us? Healthcare now, worldwide, we are started collecting patient data and all the diseases, but still there is a problem, ethical problem. Many people don't want to declare that they have a particular cancer or particular disease, and they don't want to submit data. It happens everywhere in the world, except one place I know in South Africa. The government gives everything free treatment, and only one small condition, they give all data to the researcher. They have a huge 30 years of rich data in South Africa, where I was working for my postdoc, and we are about to mine a lot of things. But anyway, I have to come back to NTU and other places after that. So that is a ethical issue is there in getting data. That is a one small problem is there. But if we get data, but now COVID data, clinical trials are in a huge collections are coming up, and we get more data. So if we get more data, then we are able to do something. Means we built a model whether it is going to be Asian problem or American problem, or is it continental based or race based, and things like that. We can mine it further and pinpoint to a level where we are able to know what is happening, and even the treatment of diabetes or chronic disease, heart disease, cancer condition. All these can be mapped in not only big data, and so we are able to understand the thing, what is happening. So for healthcare, it will be more easy once we get more data, but I am cautioning we need to get a clear, proper data. That is a important thing before we start mining things. So we are able to first identify proper treatment for a particular set of people. So it can be like a personalized way of treatment, and also reduce cost in treatment methodology, and even a lot of people getting cancer only at the fourth degree level. By the time the traditional method, we are unable to find or prove that they have a cancer. See, this is also a problem. So, but still we are having less cancer data to model to see whether we say a patient is having a cancer problem in the initial stage, the first stage or second stage of cancer, that tend to help them come out from the disease in a much better, or at least they have a instead of dying in years, they can die in 1 or 2 decades, something like that. So these are things going to happen, and things are happening, but very slowly, because we are unable to get a lot of good data, and then we are able to prevent such diseases, and we can prevent disability in many people, and we can reduce death. Death by disease is really a causing problem everywhere in the world. So are we able to do something on it? As a bioinformatics, young buddies, you can see that you are able to do something to make a better model and see we are able to fulfill this. So this is the fourth one. It's a preventive thing. So manufacturers are looking for a cost-effective. Hello, is that cost-effective medicine? Because nowadays, like a COVID.
Sir, Dr. Sundar, by mistake, you are muted. Please unmute.
Okay,
yourself.
I unmuted.
Thank you. Thank you. Thank you so much.
Yeah, okay, I unmuted. Can you hear me? Okay.
Yes, sir. Yes, sir. You are audible now.
So the next
Thank
stage
you.
is, yeah, okay. Next is, okay, this is a start slide. Preventive and personalized medicine. So once my genome is done, if I can carry with me. So what is the next step? Next step is I want to go in for a personalized medicine, means not the common medicine. I can ask my manufacturer, can you get some medicine more suitable to me or a group of people? So that is the thing we are going on. Maybe in another 1 or 2 decades, we can see that we'll have a personalized medicine, we can order it and it's manufactured and we get it in a couple of days. It's all not imagination. It's going to happen, but when, we are not sure when it is going to happen. It all depending on how fast you can have clean data. That's the main lagging now. So even pharmaceutical, the manufacturer, they are nowadays going in for silico model for potential. They bring out the structure of disease and also look for the target for potential drugs. So this one developing target drugs more efficiently in a much faster scale than on the traditional method. Traditional method is going for choosing one option, and if it fails, it goes for second option and things goes on. We don't know when they are going to hit the right target drug. But silico gives, it doesn't matter. We can run, we can add more computing power, we can run, and still it is possible to bring out something which are almost 70 to 80% close to, and then we are able to build on it. Okay, next slide. First, next slide. Yeah. Okay, here comes our topic, deep learning and machine learning. So now we have, what are the impacts happening on the big data on bioinformatics, and last but not least is the deep learning and machine learning. What is that machine learning doing here? So once I able to understand the how the genes, what are the prediction of function. So if I know things clearly, experimentally, but even though I know only for certain portion of genes, can I do something so that whatever knowledge I have, I'm passing to a person called a computer to build a model which will enable to find other things which are not known. That's where the machine learning comes into picture. We'll have some idea on the next 6 days about how things are happening on the machine learning and what deep learning is about. So the only difference between these two is machine learning means I know 10% of genes function. Can I predict 90%? That's what Prashanth was also talking. We only have 8% and 92% we don't know. But based on the data, if based on a particular set of data, if I know 8 to 10%, can I know the 90%, which is beyond our capability because of expense and funding and things? That's where the machine learning comes into picture. During my PhD also, I took some of the protein function. I took the, then we built through microarray functions, and we found it is possible to predict another 30% of data for a possible prediction for the protein function. So similarly, but only thing I told in the beginning, the microarray data are not reliable, not very clean. So that is what in the second part I'm going to tell. If I predict that this gene is going to do this function, but nobody is sure unless it is experimentally proven later on. But what is the advantage? So if a machine learning predict a particular set of genes going for a particular set of function, then we don't need to search for the gene in all the possible prediction model. We can confine or restrict only to a set of prediction possibilities, so that we are able to not search a small fish in an ocean, right? We can search it in a river. It's a more probability that we are going to hit it, or we are going to quickly find the function. That is a thing the machine learning is able to provide us. Then what is deep learning? For a particular disease or a particular function, I don't know anything. I have no idea. But if I know, like for example, if I know model in one particular type of cancer, can I use this knowledge and predict some other disease? That is called the deep learning. Means if I know knowledge in one domain or one particular disease, but for other disease, I don't have any knowledge. I cannot model anything. With any data I use, it's not coming up very well. So can I use this knowledge somehow and do it in deep learning? So deep learning goes in bit advanced way, in which we are able to use the other domain knowledge. For example, simply we say one disease knowledge, use it in the second disease, and we are able to predict something. Yes, it is possible. Some of the things were proven and things are still under research. It is possible to use the deep learning knowledge, and we are able to reduce the space. That the idea is, from the big data, can we reduce it to a reasonable way where we are able to analyze it more, better way of understanding, and analytics can be done, and experimentally we can see whether we are able to do at lower cost, rather than we do it in an open way that it's going to be a difficult process. Okay, all these are, I put the link in the below. If anyone wants to read, you can go through it, because there are a lot of literature and things are going on with big data and a lot of examples. I've just taken few to give an idea where are the fields and things is happening. Okay, next slide. Okay, I have here given some more things, like some of the thing I found. They are using machine learning models with graph-based architecture. But some of you, if you have not studied the machine learning and not doing it, some of these may be very like nightmare. It's very difficult to understand things, but it doesn't matter. The only thing is simple layman's term. I have data. Then machine learning helps in telling which are the part of data I'll be able to use so that it can be useful in my predicting something from what I know already. It's very simple like that. So that is the basic concept. And we will see during the workshop some ideas how things are happening. And tomorrow we are going to see databases, how we are creating databases, and how we can publish a database. We can put it as a web server, and also we can publish a lot of things we are going to see day by day. But all it will be 7 days, will not be enough to go in much detail. But we will see how much we are able to teach, how much you are able to grab. And the main thing is projects. I'm keen to see people coming out with interest in project, so that we can plan. That is where you are able to, once you do things, then you are able to learn things in a better way. Okay, next slide. Okay, now is the second part. Okay, any questions so far? And the different way of big data being collected and used. So far you have seen the green side. Now we are going to see the red side, alert side. The other side is challenges. When I say big data, do this, that, like beyond your somebody's imagination. But there are challenges also. It's not all that very easy. When I give 2 terabyte of data and say, can you say something? It's not so easy. We'll see how things are going to fail or how we are going to trim it. First, business analytics. It will fail to provide insights. Sometimes I cannot, if I give 1 terabyte of genomic data and see how things are correlated or between 2 species or things, finally, I may not be able to come out anything really. Why? Because of a lot of things. Lack of data. You know, even in some of the clinical data I went through, right? People don't fill up a lot of things because of shy. They don't put age. They don't put lot of things they don't fill up. They don't want even fill up whether gender, male or female or anything. Because a lot of things slowly they don't fill up. We don't get complete data. That is one part. Second thing is biased data. Like I say, like if you use a microarray data, you cannot be very sure whether it is same expression is possible in a particular gene in a particular experiment because it keeps varying. But now the technology have evolved better. Now we are getting a much better and cleaner data. So if the data is biased, I cannot do anything. Very simple. Because whatever the machine learning cannot do anything if the data is not very clean. Sometimes we don't get the updates very often. It delays process. So I expect something to get quick data from clinical trial or for some patient, but I don't get it in time. So I have to wait until I get it clean data or complete data so that I can add it to a model and then see whether we are able to do something. And traditional expectation, like traditionally we used to do analytics in a simple way. Like we can see on a classic example of Prashanth handwritten thing. It is really correct, more correct, even though it took several days, day and night time for him to pinpoint the interaction and write it in pencil or it was a traditional, but that we cannot expect the same thing in new models. We cannot say that miracle that Prashanth, you did in 2005 so difficult. I can do it in 2 days. So that doesn't happen. That doesn't happen because a lot of criteria, inherent problems are there. Data is insufficient to build a proper prediction model. So expert knowledge is not embedded to the model because many places people don't bring the expert knowledge. Means we have involved doctor when we are doing disease modeling. We are doing straight with the doctors who are dealing with it. Model carries diseases for young children less than 3 years. We're able to build model very well. Then we talk to the doctor and see whether whatever I say, is it meaningful or not any sense. He say is correct. It's really correct. Because we need the expert to add into the model. If we don't add, then things may go wrong or beyond our understanding. Okay, next. Okay, next, inaccurate analytics means we are unable to get the insight because similar poor quality of data, system flow errors. System may not be able to function properly because of the data. And finally, once I model, it is not accurate, low accuracy. Only 10% I can say this gene can bring this protein function or this person can have this disease, only 5%, 10%. Then we cannot even be sure, right? Unless it is 70%. We cannot say this group of people are likely to have this disease. If I don't get the proper accuracy, or we cannot confirm that it's going to happen. And model is not consistent with predictions. When I change that portion of data, then I see the model doesn't correlate with the prediction. The same set of people are not in the disease model. I mean, it's not very clear with the data. The model is unable to get the same result if I change, like for example, if you know the machine learning algorithm, if I use SVM or neural network or support, when I change the algorithm, then everything changes. This set of same people are not consistent. I cannot come to a conclusion whether what I am predicting is correct or not. This because data to disease could not be balanced. As I mentioned, so we cannot say the same data predict for a particular disease or not. Why? Because of if the data is not incomplete or it doesn't match a particular set of algorithm, it's very difficult to model. Okay, next. So sometimes the data, big data is complicated. Why? If I go for visualization, then if we have huge data, then the visualization also is very complicated, right? Wait, the computer can put everything on one screen, but we cannot see anything. Is it a dot or line or anything? Because the more we data use, it also get complicated. We cannot visualize it properly and say how things are happening. Even for protein structure, I cannot see a structure because the data is too much. It doesn't give a proper structure. It gives like a dotted images totally on my screen. Then that also is a big problem. The more data also causes problem, and the more we use data, and the model could bias also. So what is the involving external domain expert to decide on data and rebuild data with sub data. I don't use all the data. I try to use some portion of data and then see whether we are able to model it. Then we go step by step. That's what we did for the Cardiff model also. We divided it into 6 parts and then we used it one by one. And then because when we use everything, the model flopped. It cannot predict and say whether the children going to have caries or not. But when we break it into pieces and then we use it in a knowledgeable way, set by set, then we are able to see it's modeling very well. So the blue one, we need to solve it with external domain expert and rebuild the model with usable subdata. Next slide. So long system response. This happened many times to me also when I was going through 400 plus microarray data. Very often next day I come and see my system crash because I think you can hear me, right?
Yes, sir.
Hello, can you hear
we
me?
can hear you.
Yeah, we can.
Okay. Because somebody say they are not able to hear. Okay. System level, what happened? If I use a big data and use more data, I download every genome available onto my computer because I got a 3 terabyte machine or a very huge hard disk. Then I run something. Next day I don't see miracle. I see only the system crash. System cannot run because of memory and other issues. And also because of system delay in getting the knowledge or getting the right thing I want to use, becoming a very bottleneck. And the volume of data to be used to be not in order because I use whichever is not unclean or useless data, then I use the useful data, but system is unable to take into and process the entire data, all the set of data. So the better idea is to have a proper infrastructure and resource utilization, means, and then use the upscaling. Means I use with an expert knowledge, I use this portion of data first, then I go for the other data. Then it's more sensible that it's able to build because system also has a bottleneck, right? After some time, the computer cannot have a memory, it will crash and things will happen on that way. So it's a good idea. We use data, which is going to be how we do this. We will explain later in subsequent sessions. So we use particular data, which we know that which is going to be more useful in building our model or building our study. Then we add the other data. Like for example, I rank 10 to 1. Then what I use? I use the first rank, the more quality one, then the 2, 3. If the system doesn't crash, slowly I add. So like this, if you do the upscale or data augment, then it is possible to solve this system crash problem. Next slide. So if you use a big data, the constraint also comes huge storage requirement. You keep on adding storage, and then infrastructure cost to keep them, maintain them, and this is a hardware level is a very big bottleneck, because many labs and institutions I found they had a web server, later they don't have funds and that web server no more is working. Because of this, several thousands of biological databases always go to dormant, means it is never refreshed or not because of funding. They cannot, lot of things happen. So we are unable to get those data, clean data, somewhere lying because of the computer problem, or it is they don't have funding to renew the web portal. It's all lying. Even I know in many places we worked now, all the 10 web servers we developed in South Africa, all is now dormant because of some problem. The server crashed and it is still crashed. It has never come up, even though all things are published, but we don't have a backup now. Anyway, these are things happen because of expensive maintenance. So what are the things are good now? We can have cloud solutions. Now Amazon, Azure, a lot of cloud solutions are coming where you can pay as per your use. Even government and all, we are using cloud solution, because when I want to scale up, I pay only whatever amount I am scaling up. When I scale down, I pay the normal one. So like this, we are able to use a simpler version and scale up methodology where we are able to cost save and also utilize wherever we want, we are able to do it. Okay, next. So we have seen how the big data is like miraculously very nice, that I had a lot of data I can do in various domains. But at the same time, the myth, the challenges, it's not that we just include everything and we are able to get something. It doesn't happen. There is no machine learning robots which decide what I have to add and what not, and then finally come with the solution and say, okay, this is your answer. It doesn't happen. It happens in only fictions and movies, but still we are yet to go into that level of possibility. But one thing I told in the beginning, I worked in biological, in various domains, even in banking, I used all the bioinformatics tools. So we have used, so when you learn bioinformatics tools in a clear with projects, then life will be very smooth for each of you. Those who are doing masters and later you want to do PhD and things like that. You can do a lot of things. You can model anything in the world. But again, caution is you need a clearer data, and also you should know how much you have to use for building a model for prediction through machine learning or deep learning or anything. That is the thing as a caution. Okay, now we can take some questions.
Can I ask one question, Dr. Vijay?
Yes.
So you said that micro error data is biased and it is not very reliable. So in what, in what sense it is biased?
Because the same microarray, earlier I'm telling, because currently I don't know. Earlier, when we used to get full scale genome, right? The good point of microarray is in one shot, you can get all the, I was using equally. So 7,000 genes, it gives all the expression values to a particular experiment in one go. But if you do the same experiment one more time, you may get different values for expression.
Okay.
So it's not consistent. So later you cannot say I build a model and you another microarray and then you can predict it properly. It may or may not work for any particular, because some protein it happens. From some function, somehow I cannot predict it. I cannot model it because the data is very vast and it is not properly happening, because either the gene should express high or low, right? Like a statistically, I say this set of genes should have a high expression value. That is expected. But next time when you do experiment, it may some go less. So if it goes like this, we cannot say sure that this experiment, whether it is going to be high or low. Even in statistically, we cannot do anything, right? It will not be a proper way of representation. But if you put it to the model, even, machine learning all is basically from statistics, started with statistics, but it expanded more with algorithm based and things like that. So if the base is not working properly, computer also cannot do anything, right? So if we use a biased data, then you cannot go anywhere. You cannot predict or you cannot model with set of data and say with the high expression, this is going to be the protein function.
Okay.
If we can just interrupt, I mean, that was wonderfully inserted by Sundarda. One of the other things is, in case of microarrays, you will not be able to probably, identify the whole set of, transcripts or genes, through hybridization. So, whereas in RNA-seq, you will have a full compendium of it. And although, microarray is more sensitive, you cannot really get that enriched gene expression data that is amenable, for machine learning. That is what, Sundarda's point is. So the other way around is, if we have RNA-seq data that is highly translatable, unlike the microarray data, and the RNA-seq data, would have an enriched something like, fragments per kilobases per million reads, where we'll know how many number of times a particular transcript is highly expressed, and then we'll have a divisive count, and that particular divisive count can be better used for machine learning heuristics. So that is the bottom line. Yeah.
Right. Thank you. And so for deep learning, again, you need a template kind of thing. So if something is very new and nothing is known about it, so how to go about it?
Yeah, we need to somewhere to the knowledge, right? If I say disease, then disease to disease, it may be workout, right? Because disease to disease, you may have similar set of data. That is the important thing. We don't have similar set of data. Maybe the data points are not the same. No, experiments are not the same. But still, if it is totally deviated from data to data, then it is very difficult, right? Because the domain knowledge, means the set of data should have a similar points, means what the collection of data about it. If it is total for disease, I cannot model to protein. You got my point, right? Because it's not be correlated between the data, because this is a clinical trial and it is a sequence data. So that I don't think anyone has tried on that way, because the idea is with the knowledge of machine learning, it is possible to embed it into a layer so that the other model can inherit it and then it can go on with a similar set of data, similar kind of data. We can say not the same set of data, but kind of data.
Right.
Dr. Sundar, can we take few queries from the chat box? So we have one query. Is there a difference between artificial intelligence and machine learning?
Okay. Is it something like one and the same? Machine learning, when I say how the machines learn, is machine learning. Artificial intelligence uses the machine learning techniques and finding something. So as a robot, we can say is a robot, which is supposed to be having its own algorithm to find things and then bring it back. So artificial intelligence is the next layer. I mean, when I ask something, then artificial intelligence may use machine learning or inherent knowledge or deep learning or any of the other possible avenues and then bring out the answer. That is artificial intelligence, means the self-knowledge. But the self-knowledge comes from like a dictionary or a model which was already proven, something like that. So it is one between the same.
Okay, thank you. Thank you,
How
Dr.
is
Sundar.
artificial? Is because of this model, the knowledge is inherited in the artificial way. That's what we say artificial intelligence.
Can we take one or two more questions from the chat box?
I cannot see the chat box actually because
I will read it for you though. So how can we relate machine learning with pharmacology? Yeah. Yeah, Sundar, KK Shine Marma asks, how can we relate machine learning with pharmacology?
Yeah, pharmacology is for bringing the targeted drugs, right? So I told any domain where we have data and we need to know what are the data required in bringing out a targeted drug, mostly like a protein structure and how it binds with the disease, how it activates and how the disease is dissolved. This is the actions happening, right? So if we get this data and we are able to decode this data in a tabular form in such a way, we don't put a protein picture and say, can the machine learning do something? It cannot do anything, right? It is just an image. So we have to decode the image or structure, everything into a table format and bring like location or number of atoms or whatever you call. So then I correlate as a table, like the table may have thousands of columns, which brings the characteristic of what you see a picture or what the pharmacology people do the binding and things. That information can be modeled. Then we can use machine learning for pharmacology, how the targeted drug, and this is actually one project we are already doing on it. Some of you will be surprised that we are having children bioinformatics group, and really they are bringing out very good projects, the children, and one they are doing this particular protein model, which is also like a binding and things they're bringing out, and we are still working on it, and we are going to move it to see how we are able to use the machine learning and things like that. And other group of children, they are using a COVID database. Actually, they come out tomorrow. I'll see if they are having exam. They can show up their face for a few minutes, how they prepared the COVID database. They are only grade 8 children only. So any domain is possible to use. Only question is, do I have enough data, clean data? Is it? Then we go for the machine algorithm and see whether it is going to use it systematic way, and then we are able to predict or model it. Then we can use it in any domain, not only in pharmacology, because I have used in most of the domains. Yeah. Any other question?
Yeah. Yeah, please go ahead, Rakesh. Yeah, please, sorry. Yeah.
Okay. Yeah, it's from Madam Anchal Bharati. So the query is, can we replace experiments on living organisms for drug trial with the help of bioinformatics?
I told you, right? Some extent only we can do. We cannot do 100%. That stage is yet to come because we are yet to get a clean, complete data in any domain, whether it is structure or sequencing. And also I told, if you get 5% knowledge, and are we able to predict 95%? Is not that easy, right? Because if I know 70% in my class, they're all intelligent, then I can pose question. Only 5% are intelligent and pose difficult question. I cannot expect that they can solve it, right? Same thing, it goes to the machine learning also. If only we get better knowledge, I told in the sometime, maybe in after 1 or 2 decades, it's possible everything is done in silico. Everything could be possibly done, but still people are having ethical issues and other some problem or other. They are not releasing the complete clean data. That is a big problem now. Unless we get full data, yeah, it's possible. In future, we don't need to wait for a very long years to get a drug or anything. It's going to be happen very as soon as possible. Like that's what I told, like personalized medicine. I see a doctor today, and in 2 days I get my personalized medicine only for me, just to make me recover from my illness, the level in which I am in trouble, something like that, not the common medicine. Is that okay? Got the point?
Yeah, I hope that the participant may have got the response. We have 2 more queries. Can we take that?
Yeah, quickly we can take.
Yeah, actually the time is also, session is going to be over. So I think we can take 1 or 2. The other query is from Ms. Neha. Can biased data be transformed to clean data?
Yeah, it's possible. Maybe if she wants to take this project, we can work on it. Because it's also a thing how the data is biased. If we can find out how it is biased, where it is problem, then it is possible. Yeah. The only thing is the knowledge is required. If Sneha gives knowledge how it is biased, then we can build the model. Then other bias can be found out.
Okay, thank you, Dr. Sundar. So I think I will have the last question of the session, so we can then go for conclusions. So that is, if bioinformatics has challenges like inaccurate insight, unable to fit big data, and also it is expensive, why we are still using current existing tools?
Because existing tools are even more expensive. If you go full-fledged traditional way, it's beyond our anybody's budget. Now budget even reducing day by day, and COVID has reduced 70% of budget. So how we are going to go for the traditional way in which we are going to use NMR or finding things, it's beyond our imagination, right? So now Silico only is able to solve our problem. But still we have to fight. That's the thing. The data may be clean. We have to make a trial and error methodology in which we have to, if we cannot use 100% of data, the good point is I can use 70% of my data and see how much is, because it's possible in machine learning to remove the biased data. It's possible to not take into the model all those data which are conflicting. Even in statistically, we can say these rows give not a proper correlation or something. If you take a correlation analysis, you can see these particular rows doesn't go in a proper way because all the other rows correlated to 1-to-1 upscale or downscale. But some rows, maybe 10-15%, doesn't follow that rule. Just we remove that. That's all. We don't include them in our modeling methodology because there is always possibility of biasness 10 to 15 or 20%, depending on the data.
Okay, thank you, Dr. Sundar. Thank you very much. I think
yeah. Finally, I have
more queries
place, because yeah.
Yes.
So you can post it. Yeah, students can post the questions, and we will see on the next 6 days. We will try to address some of the things.
Yeah, right. May I request Dr. Nisha to proceed further?
Sir, if this is all, then we can conclude today's session. I would like to thank you, Dr. Vijay and Dr. Prashanth, for the informative talks. And we can meet tomorrow again at 5 p.m. in the evening. Thank you very much, sir. Thank you very much.
No problem.
Thank you.
Thank you all for your patience, students. So you don't feel that you are studying bioinformatics, because many people sometime back they used to tell, I'm studying bioinformatics, because the only subject I got it, or the only module I got. But now you should be proud because I've used it in all domains. So it's possible to use it, but you need to know with experience. That's the main concern. Okay, we'll go through it. down the line. Okay, see you all tomorrow.
Thank you,
Okay,
sir.
thank you, Dr. Sundar, and thank you, Dr. Prash. So we'll meet tomorrow.
Bye.
Till
Thank
then,
you.
take
Thank
care.
you.
Bye-bye.
Thank you very much. Bye-bye.
Thank
Yeah.
you.
Bye bye, Prash. Thank you for, yeah, tomorrow let us see the internet may be okay.
Okay. Thank you. Thank you.
Thank you. Bye bye.